Welcome to this beautiful product building on our campus. It's part of the new building at the University of Arkansas. Those of you who are in Zoom and may not be in Arkansas, it's a gorgeous day today.
Yeah.
And as I was driving in, the daffodils were peeking up, and I'm like, oh, I need to get these daffodils for the home and for the office. Yeah, so it is the beautiful spring is upon us, and let's hope no more dips in the temperature, and we are pulling out our down coats, So my name is Ranu Jang. I'm the Associate Vice Chancellor here, but most importantly, the Director of the Institute for Integrative and Innovative Research, a mouthful, but we call it I3R. So I think many of you know about I3R, but every time there are some people who are on Zoom or on in person who don't quite know about us. So we are an institute that is at the university, across the entire university. We were formed with a transformational gift. And our mission is to pioneer solutions to wicked problems. And how do we do that? We do that through convergence research, basically converging not just ideas, but sectors, different sectors together, to find solutions that may come out of by bringing together our academic world, of course, but also industry, government, community partners, all of those together to be able to come up with answers to problems that are not easily solvable and don't have a single answer and require us to take risks. So that is part and parcel of our DNA, but we are very mission driven. And our purpose, really, the big purpose is societal impact. And then I was having a discussion yesterday, and somebody said, societal impact is so big, and how are you going to ever do all this? I said, well, each one of us can do some aspect, keep that in mind, and then we will make a difference. And we believe in that passion of making a difference. We want to be leaders in this convergence research approach, and we want to be a preeminent partner to be able to deploy solutions that actually go out into society. And we've said, okay, we'll take on a grand challenge, and our initial grand challenge is integrative health. It's not health as saying it's a health institute, but integrative health. What do you eat? What do you exercise? Are you getting your sleep? Some of us are not getting it right now, right? But then it's really about the well-being of everybody. And so today's mini symposium is really talking about a wicked problem. It's addressing part of what we can do to assure a good health and well-being for all our peoples, but really for a global population as you think into the future, and you're getting 8 billion people, and we have global economic growth that is needed, and we need demand for protein. And so there are many different ways that could be done. But we definitely know we need to have a sustainable solution. And some of it may be out of the way, not the traditional ways of addressing the growth of that protein production, but maybe new ways in which we might be thinking about how we might do that. So alternative proteins is one way in which we might address this issue. And so today what we are going to be doing is, it's really a whole day, and we're kicking off this morning, but we're going to be talking about the basics about it, so learning, getting educated about it, but also then we'll be discussing, and you want to ask our speakers today, what might be economic issues, what might be cultural issues, what might be social hurdles, what might be many kinds of other things that might come about as you think of a new way of doing something. Some of it is not quite new, but it may be new how it is integrated all together. So we have 2 fantastic speakers to start the day for us. And we will kick this off. We intentionally picked 2 people to get to the discussion, to bring 2 different approaches for all proteins together. And one of them is the first talk is going to be by Professor David Kaplan. Professor Kaplan is a distinguished professor of engineering and biology. He holds a Stern Family Professorship at Tufts University. Tufts University has a Center for Cellular Agriculture, which is supported by the USDA. Nobody could be better suited to lead it than Professor Kaplan. He's a member of the National Academy of Engineers, National Academy of Inventors, but most importantly, he has a passion for making sure that students and the future generation are prepared to lead our future. And over the years, I've known him because of the biomedical side of tissue engineering that he comes from, but he brings this drive, this passion to this another big aspect where really you are merging two things together. So we are very fortunate to have Professor Kaplan with us. Second person who will be talking, we are not going to have a gap for questions in between. We're going to go straight to is to Dr. Sun Ferrer. So Sungil is recently come to the University of Arkansas. So many people may not know him because he's just landed here, but he comes in the Department of Food Sciences. And you all know we have a great ag division too, and we have bumper scholarship. I see one of the chairs of biosystems and ag engineering here. And Sun also spent time at a startup looking at all proteins. And today he's going to talk to us also about mycelium and fermentation and how that gets pulled together with the cellular meats and agricultural byproducts. So two very interesting talks. We will learn, we will get educated, and then we will open it up for question and answers to both of them. But this is our opportunity to really learn. And so please make sure any questions you have, any questions you didn't have but come up, no question is an odd question to ask because many of us are just new to the space. We are learning about it. And so here we go. It's going to be a long and fun day. Professor Kaplan.
Thank you. Thanks. So now my hour talk is only 40. We love you anyway. So first of all, thank you all for coming out this morning. I really appreciate it. I'm really excited. My first trip to Fayetteville, so I learned a lot yesterday. And my Uber driver Wednesday, late Wednesday night, taught me a lot about the city. So this is really a lot of fun. I had a great day yesterday, and believe me, compared to 30 degrees and snow in Boston, this is a pleasure. So thanks for having me, number one. I have kind of a tall task in my limited time to talk to you. I'm going to kind of divide it into threes. The first part I'm supposed to do like cell ag, sustainable proteins 101. So back to school for many of you, even though you're faculty and others. So I'm just going to, apologies to Eric and others in the room. I mean, you could recite this chapter and verse already, but so bear with me on that part. And then part two will be a little science because it's just my life, right? So I have to tell you a little bit about what we do in the lab to propel this field forward, or at least we hope to, and just give you a sense of where we are, where the challenges are, and hopefully get some of you, including, I know there's a few students here, excited about getting involved in this field. We need the students to drive this forward. And then the last part, I want to tell you a little bit about what we actually do in our program, more programmatic efforts, and hopefully how that might lead to some synergies with your plans here for the program. So that's my goal. I call this the road, this is silly, I know, road from sustainable proteins to tissue engineering to cell, ag, or food of the future. And the reason is this is the history, my life in one slide, which is that's the way it is, right? So the lab has always been founded, even my PhD, way back in the early days, right? It was based on biopolymers. So I always cared, don't ask me why, it's in the DNA, about how nature uses interesting materials to solve complex problems, right? So I studied actually lignin biosynthesis for my PhD, just so you understand where my crazy brain comes from, right? And so we have a large program over decades to study what I call fibrous proteins, which are really the early day sustainable proteins, things like silks, keratins, collagens. These are the things that I've always enjoyed working on. And we work on those from taking natural materials and kind of repurposing them using engineering into new materials, to using genetic engineering to sort of reprogram the DNA and make polymers that don't exist, but do new things to drive new properties. And over the years, that's led to the spin out of, I think we're more than 15 startup companies over the years, many licenses. We have medical devices and products from these activities in use today. So it's kind of rewarding to see that field mature from our lab. And that led back in the early, I guess we started in 2004, we were funded by the NIH to move into the field of tissue engineering because of our sustainable biopolymer engineering work. And so we've had what's called a P41, if those of you know the NIH program, it's a resource center on for ours is tissue engineering, and we're still doing it today. I was the PI for 15 years. You have to renew every 5 years. And now my co-collaborator on this, who's at Columbia University, is the lead PI, and I'm the co-I. So we continue to develop those kinds of activities. And then I put in here because it may not be obvious, but I'll tell you a little bit. So we've had a U19, and also we now have a brand new Department of Defense MURI on our intestine tissue and our brain tissue. So these are human tissues we grow in the lab. And I put them here because, in fact, the U19 on intestine tissue was to study pathogens in your gut and what they do. But now we're starting to use that to study the foods we're making and how they're absorbed in the gut, what they do for inflammation and so on. Think about therapeutic foods and so on. And again, happy to discuss all these things. And the MURI on the brain is a little bizarre, but if you've never thought about it, sleep tight tonight, right? Because in fact, there's this whole new emerging field of actually microorganisms or viruses, from COVID, or parts thereof that end up in your brain. And there are many paths to get there we can talk about. But what do they do for your thinking and so on? And all this in terms of gut-brain signaling relates to human health. So the idea of talking about foods here today all relates to this theme. So that's a long-winded path to get me to what we're going to talk about today, which is cellular agriculture. We started in earnest in 2016. We actually started 10 years before that with some small things, but nonprofits out there, New Harvest, GFI, were able to support our students early on, and that led to our National Institute in 2021, funded by the Department of Agriculture. And I'll talk a little bit about that at the end if I have time and don't keep talking too much. And we have our own center now, and again, we're hoping to build some partnerships with the group here as things evolve. All right, so that's history. Now 101, right? So I want to talk about future foods, cell ag. This is my attempt to explain what it is. It's essentially growing meats and foods without the animal, right? All we need are the cells from the animal, one-time biopsy. We never need an animal again. That's the holy grail for this field. And we'll talk about why. We'll talk about these. And these slides are not my words. These are from your own Department of Agriculture. So don't blame me, okay? But this is what we're facing. And Randall mentioned it already, right? We're going to have 2 billion more people on the planet in the next 20, 25-ish years. And according to the Department of Agriculture, we need more food, more water, more energy to feed that extra population. And our current ag system is not equipped to meet the demands. That's from their own cell ag reports. I don't know what the farming community is like around here, but the other issue that we've come up with is that the Department of Agriculture talks about is that, we peaked in farming in 1935, 7 million farms. Roughly last year or the year before, we were down to 2 million. You could see the precipitous drop in farms, right? And it's not just the loss of farms and farmlands, but the vast majority of farmers can't survive just farming the way they do, small and mid-sized farmers. So they have to supplement income, and it's a real problem. So one of the holy grails for what we're talking about this morning is, can we find ways to supplement farmers' income in new ways that they haven't been able to have access to before? So that's, again, one of the major goals here, and that's why the USDA has funded our institute to look at this. So innovation is key. And again, these are not my words. This is Department of Agriculture. We have to innovate. We have to look at alternative novel foods, protein sources, cell ag, biotech approaches to try and fill that gap. And how are we going to do it? No one has the answer. This is what I'm talking about is one of many options moving forward. And we need to do it soon. We can't wait another 10, 20 years to get started. So that's your context, if I could call it that. The other is this, and you all know this. Meat consumption around the world continues to go up, despite every attempt to slow it down. Countries get more well-off economically, they want to consume more meat. There's a direct correlation, US as well. So that's not going away. So the question is, how do we really focus on generating more meat to fill the void? And that's really what I'm talking about today. And just one reference point, not to dwell on this if you're not aware of it, to make a quarter-pounder hamburger that you get at McDonald's or somewhere, right? It takes, almost 7 pounds of grain and forage. The cow is the model of inefficiency, right? We love the food, but to get it, it's very costly economically and resource-wise. So these are all things to worry. So this is a summary of all the drivers. And what I love about this field, and those of you who are faculty, right? In my decades of doing research on materials, tissue engineering, I have never had more passionate students involved in my lab than this, by far, right? They're just passionate. And they're passionate because they either care about food equity, you know, how to make sure we don't have food deserts and things like this. They care about animal welfare. They care about the environment and sustainability. They care about just how we're going to feed the people I just mentioned. And they also care about food safety, food security, which is becoming a major issue not only in the US, but around the world. So these are all reasons to get involved. What I like is you can pick any of these points and you become a sort of a real participant in the process. And we need everybody, right, to make this go. So this is sort of the driving force behind this. Okay, now the 101 really starts. So I'm sure most of you know about these already, right? These are plant-based meat substitutes, right? Whether it's the Impossible Burger, chicken nuggets, these are good examples of what's out there today. For those of you who followed the field, like Eric and I, you know, we went from zero interest in these products to like in every restaurant, everywhere you go, you find these products today, which is remarkable. And the magic ingredient that I think propelled the field forward was this, well, last night we talked about it's not the only magic bullet, but this ability to incorporate recombinant heme protein with iron in these plant products, give it that metallic taste, which is meaty, you cook it and you want to eat it, right? So that's really what drove, I would say, a lot of the sort of real growth of this field. And there's other examples that are not meat-based, but, you know, mung bean protein, if anybody's tried just egg substitutes, they're not too bad, right? But these are other kinds of proteins out there people are making into food substitutes. So this is the cartoon of what that all looks like, right? We take our favorite plant, corn is shown here, it could be anything, the mung bean. We somehow figure out how to chop it up, mix it, degrade it, whatever you want to do to get it into a form factor we can process. It's all about processing. We then go through some extrusion or other way to make it into something else, and out the other end comes an extruded product that has the right features, et cetera, for a consumer to want to buy it. So this field, plant-based production, took off tremendously, and then as you all probably know, it's kind of like all of a sudden went up and up, and then over 3 to 5 years, it's now crashing. And the reasons are many. My take, and I think Eric has his own, are one is cost is still a little bit of an issue. Taste is still a major issue, even with the heme in there, it's not up to the standard of meat. And number 3, there's a lot of some additives that go in to make the process work, and a lot of people don't want over-processed food products anymore. So these are all possible reasons why this field is not doing well. So this is where plant-based are. So you have that in the right context, and I'll put them all together when I get to cell ag. So the second thing is just alternative proteins, period, right? So growing proteins or making proteins to use as an ingredient, really not as a standalone food, but as an ingredient in foods. Good example of Perfect Day animal-free milk, right? So they produce the casein protein in large bioreactors, add it in and create these products that people want. They make yogurts and cheese, and if you've ever had their ice cream, it's really probably one of the best ice creams I've ever had. It's really good. But they're also crashing and burning, and they're crashing and burning because of the cost. It's very costly still to make these products, but it's not that different than things we're all used to eating and consuming, right? It's just a different variation on a theme. So this is the cartoon of all that, right? Take your favorite gene, whatever it is, whatever protein you want to make, pop it into your favorite host organism, bacteria usually, yeast, et cetera. Create a large bucket of that, grow a lot of it, then isolate downstream as we go to the right, purify that protein, and then use it as the ingredient. Again, the major impediment remains cost. How do we bring the cost down? And I think there's a lot of work going on around the country to try and look at alternative hosts and make this process less and less expensive, but we have a long way to go. So lots of opportunity there. And then we come to what I'm going to talk about as focus. So here's a couple of slides of things we've eaten during our visits to the companies, right? So here's Mission Barns out in San Francisco. There's their bacon on top of a BLT. And it's, again, what they say is real pork without the pig, right? So real pork without the pig means we have pork fat cells, and those are an ingredient into a plant matrix to give you the pork taste and flavor, and the texture comes from the plant protein. And here are their meatballs in here. Okay, same idea. pork fat cells, et cetera. Here's sci-fi foods that make beef-related outcomes. There's a hamburger we fried. And here's wild type, again, out in San Francisco. They make salmon by cultivating cells from the salmon and adding in some other ingredients, right? And that's if you prefer rolls, you can do this. So anyway, we've tried them all. They're really not bad, but they're not yet really good, right? There's still work to be done. This is a simple summary. I'm happy to talk about more of the companies as well. So this is the way we present cell ag, if you're not aware of this field, right? So take your favorite animal. Cow is shown here. We go out to our vet school at Tufts. We take a biopsy of the animal, isolate stem cells. They may be muscle stem cells, fat stem cells, other cells. We then, in the middle there, grow a few cells into a gazillion cells. You need a lot of cells because they are the component we're going to use. They're the taste, they're the flavor, they're the nutrition. And then we go on there to the right and we start to have some kind of biomaterial matrix onto which the cells are going to live. So you get texture and all that sort of mouthfeel at the same time you're getting the taste and flavor from the cells. And then most goals are to make products that consumers already know as form factors they recognize, so they'll at least try the products. So that's kind of the process I want to talk about in my part two. All this field came out of tissue engineering. Remember, I told you my career was sort of biopolymer engineering, biopolymers, sustainable proteins. Then we moved into tissue engineering. So we had long studied clinically related human tissue growth in the lab. I mentioned our brain work and our intestine work. And really, once those tools are here, right, the right cells, stem cells usually, the right matrices to grow, to host the cells, and the right ingredients to get the cells to grow, there's not that much different between making something to repair the human body as opposed to making food on the bottom right there. The biggest difference is what? Price, cost, right? You know, people will pay virtually anything here, right? If you have a bad tissue or organ, people will not pay more than what you buy in the supermarket today. So this is the big chasm that we have to cross. And we're not crossed yet. We're working on it, and many in this room are working on it, but a lot of work still to be done. All right, and I integrate everything I've told you so far into one slide because this is the way we look at the field, right? So we talked about sustainable proteins, right? So alternative proteins, depending on what you call it. So these are the acellular products for microorganisms, ingredients. Proteins are important. We talked about the plant-derived materials. Those are acellular plant-derived products that go into texture and mechanics, right? And then cell ag, it's really the cells as the main product. And then we put that all together here, right? Left to right. Get the right cells at the top, lots of cells in the bioreactors. You've got to feed those cells. So we need the plant-derived materials as sources that we can use to create the media, and I'll talk about some of that. And that all goes in to drive the process. And then to the right, you can either make unstructured meats at the top, which are things like hamburgers, sausage, nuggets, or you can make dense structured meats at the bottom. All that needs, again, more additives from plants and more of the scaffolding from the biomaterials, alternative proteins, et cetera. So to me, this is the synergy of all those fields. They all need to work together to give us the right taste, texture, and eventually cost to drive this process forward. So let me just stop there. Any quick questions? I know I'm not supposed to ask questions yet, but I will.
What kind of questions?
Please.
Hi, Rachel Falcon from Cobb Genetics, the gene breeding company. So we spend a lot of money on developing these alternatives, these plant-based alternatives, which is great. I'm all for it. So please don't take my comment the wrong way. But we're diversifying products that are available to us, and yet back here we have the animal products, which we need to make more efficient as well. And so there's that divide of finances, of resources. Where should we be putting our money? Should it be in the development of the new stuff, or should it be making our traditional protein sources more efficient?
So my answer is yes. I don't think no, I mean that. No, I think we need to be pushing forward on both. We have to continue to make things more efficient for what we have. There's no question about it. We have to continue to look for utility for everything right now that's going to waste, that's not being used in that process, where this can come in. And then we need alternatives. We're not going to get to that 10 billion in style if we don't do something else. So
I don't disagree with you. I just, I find
so I think we need both. Great question. Thank you. Okay, I'll keep going then, but we'll have time. So if you're not aware, the field is booming, okay? We've hit some roadblocks with the economies lately, right? But if you just look at alternative proteins, you can't keep up with the number of companies in the US, around the world trying to do this. We have examples even in the room. Right? If you care about cell ag, the number of companies continues to boom. We'll see some consolidation in the next few years because of the economy, but they'll continue to grow and thrive. And then the B2B, suppliers, we've talked about that already. Lots of suppliers, we work with some as well. Lots of people in the space of making sure they're getting in line. How are you gonna make enough sugars, enough amino acids, enough growth factors? These are all things that we don't have anywhere close to the amount of materials we need to really drive this process. So, my message is get on board if you're interested. There's plenty of options to really help here as we go forward. And then Eric can speak to this because this is his baby, right? But he's been able to shepherd through the regulatory process and approval to get the first 2 products now on the market, literally available to be purchased and tried. His company, Upside Foods, in the past really was the pioneer here, and he can talk to this later. And then obviously also Good Meats is on here as well. And if you're not aware, for cellular agriculture foods and meats, chicken is the first products that were approved. They don't just go through the USDA. They also have to be approved by the FDA. So they have 2 levels of scrutiny. And so when people say, well, are these foods safe? The answer is, of course, they're safe. They've been approved, right? Just like any other food we eat. So that's my comment there, and I'll leave any further proselytizing to Eric since he's really the world's expert in this space. Okay, so it all sounds great, but it's not, okay? It's great because it's real. It's great because we need it. It's great because it has an immense opportunity to make a difference in all of our lives, but there are a lot of challenges ahead, right? That's why we need all of you guys. We need the students to roll up sleeves and help. And for today, I don't have time to go through all this, and I'm trying to keep track of time, but we have to keep bringing those costs down, as we mentioned. We have to deal with scaling of manufacturing. To me, that's the biggest unknown still today. How do we get from where we are to where we need to be? And I'll touch on each of these very briefly. The thing we only briefly talked about on the right-hand side is the dense foods. It's relatively easy to make hamburger and chicken nuggets. It's relatively almost unheard of to make a dense steak or pork chop or things we all want, right? And more than 50% of the meats consumed by the American public are dense food. So we have to get there. And I'll show you one example of one of our, we say innovative ideas, some would say crazy ideas of what we're doing. You can be the judge. Whatever we do, we have to do it safely, right? So we all follow certain rules that are already promulgated, and we have to build new rules as we go with these new kinds of processes. So that's going on. I'll show you what we're doing on food taste and quality to our discussions earlier. We're just at the beginning stages. And a lot of this, and then do you want GMO or not GMO, genetically modified foods or not? As an academic lab, we believe we provide research to look at both, right? And we look at what can we do with genetics today to get us there, and others don't want genetics. So we'll publish on both and let the consumer eventually decide and let companies decide. But I will say the approved products from Upside and Good Meats are all genetically modified, I think. So as far as I know, these are all already approved. It'll be up to consumers to decide if they want. All right, so lots to go through. So that's the end of, I think, part 1. Yeah, okay. So now you kind of have the basics, at least I hope, in terms of how I'm defining things. And we can debate about the definitions all day if you want, but that's fine. I want to go into now a little detail on what we're doing. And I'm going to walk you through the cartoon I showed you from left to right and just give you a few snippets of the research, what we've been doing and where some of the challenges are. So you have a better sense of looking under the hood and seeing what the reality is. So let's start on the cell. So this is what we do in my lab and most labs, right? Again, we go to our favorite animal. We get the biopsies. We then literally never need the animal again if we do our job right. Okay, so that's the goal. We isolate in the lab those cells. We bring those biopsies back to the lab. And we characterize the cells in terms of stemness. And then we have to figure out how to get those cells to differentiate either into muscle or fat to give us the key ingredients for these meats. And you can see an example, this is getting old now, it's from 2020 when we published it, but on the right-hand side, there are all these markers that tell us the cells are the right cells from the cow, and the merged image on the right bottom, this picture here with all the nice colors, that's a spectacular muscle bundle, right? It just means the cells are all the way down the lineage, form this cluster of cells into these fused myotubes, and it looks and feels just like a muscle. So the process works. There's no mystery from a technological standpoint of getting these cells out and making those kinds of muscle tissue, and that's the core of meat, right? We need to do that to make meat. And under the right conditions, we can show that these cells actually contract and so on, so they're functional as well. That's a long way, though, from where we need to get to. Just keep that perspective in mind. We're talking a couple of hundred microns diameter of the muscle bundles versus a slab of a steak, right? So we are nowhere here, okay, left to right. Now, there are companies out there trying with 3D printing and other methods, but it's still a challenge to make not only the dense structures, look how dense those myotubes are. That's hard to do and keep growing because the cells get shut off from supply chains. The cells die and we don't get the tissue to grow. You get necrosis. So again, I'll come back to how we're trying to overcome that in a little bit. But this is where we're at, right? We're doing great on the right. We're horrible on the left step, but lots of ideas to get there. Same with fat, right? Fat, amazing. Here's you can't tell, this is pig fat. So these, again, fat biopsy from the pig. And you can see fat cells, this sheen of kind of whitish, translucent material. That's just a layer of fat growing in a plastic dish from the lab, right? And this is all pig fat. And then we just scrape it. It's a little hard to see in the yellow disc. You can see like a bundle of that fat when we're done. And that's what we use for our smell tests and other things we'll get to later. But, these are adipose-derived stem cells grown up, differentiated into fat. And, we take it from there. And without all the details today, one of the really important factors for this field that you can't do with an animal is because you have direct access to the cells when you cultivate them in a bioreactor, you can feed them what you want. So you can feed them healthier fatty acids. You can feed them other ingredients. And the figure on the right simply says we can alter the composition of those fatty acids depending on what we fed. And this is all published now. So this is one of the really important factors of this field. You could start to dial in, let's just say, a nutritionally enhanced fat that you can't do with the animal on the hoof, right? You don't have that luxury and that control of what goes in and what comes out. So these are areas we in other labs are working on very actively. The other thing you have to worry about is, like I said, we never want to go back to the animal. So you have two choices. One is spontaneously immortalize the cells. So just keep growing the cells and eventually they mutate, self-mutate, and they just grow forever. That's the ideal case, right? Because there's no genetic manipulation we purposely do. So from a regulatory standpoint, they're perfectly fine to use. The other way, if you can't spontaneously immortalize cells, you have to go in and genetically accomplish that goal. So for our bovine muscle stem cells, we cannot yet immortalize them spontaneously. So we use genetics. The first technique we use is this. Details just briefly is called a transposon. Insert DNA in, and the red line shows you they grow and grow and grow. Those are days in culture doublings. So they just keep growing. So we've successfully immortalized those cells. And if we didn't do it, the black line shows you the cells peter out and stop growing. Just like any cell in your body stops growing due to what we call the Hayflick limit. You have a limited number of doublings before your cells go into senescence. Importantly, those cells still differentiate. Again, without details today, they still become muscle and so on. So the process works. We've now replaced the transposon approach with a CRISPR approach because it's much more precise. And so from a regulatory standpoint, it'll be a much easier path forward. But still, it leads to a lot of questions, right? Do consumers want it? We'll find out over time. And the nice things about it, number one, the speed of discovery is so much faster than not genetically modified systems. So I think the products you're seeing and you will see are all going to be genetically modified to get there faster and cheaper, but time will tell as well. In other cases, we have no problem, with the pig adipose cells, fat cells, those spontaneously immortalized, no problem, we don't need genetics there. And then we had an example of fish, and I don't know if anybody else is from New England, but if you're from New England, we got on a boat at Gloucester, Massachusetts, deep sea fishing. I actually got on the boat too. I actually got seasick too, but it's the team, so that's okay, We caught fish out there, we caught a mackerel out there, we took biopsies out there, brought them back to the lab. Very quickly, we were able to get them spontaneously immortalized, sequenced to make sure they were mackerel and all the right things. And that was licensed out to a startup company almost within 1 year of the isolation. So things can happen fast, but other things like we've been trying to isolate and immortalize lobster cells, right? We're in New England, lobster cells. We still have failed 5 years later. So it's not mad, there's a lot of magic. It's not straightforward science. There's a lot of challenges here.
Can you talk a little bit why you've observed the speed of innovation tends to be faster in this space from media development, cell line generation versus cycles?
Yeah, it's a good question, Eric. I think there's a couple of reasons. One is, it's funny because like I said, we started in the medical side, right? And almost every lab that studies stem cells and media, it's all about the medical side. No one spent time on optimizing things for costs and efficiency. It just didn't exist. You didn't need to, right? It wasn't until this field started to become a field that people started to say, oh yeah, wait, that's good. And the other thing is there's never been public money, grant money to do this, right? The grant money always goes to medicine. Ag goes to traditional ag, as it should have. And now that this field is emerging as an alternative, right, now you're starting to see some funding where it goes to labs so they can start to ask these questions. And now that that's happened, you're seeing this sort of exponential growth in all the ways to reduce costs. Because I don't think costs are going to be the issue at all. And I'll show you some examples of why I believe that. I still think scaling is the biggest challenge. And we've talked about that quite a bit yesterday. Yes, please.
I had a question on the spontaneous immortalization. You said you don't have to genetically manipulate them, but do you know what happens in the cells when they spontaneously immortalize?
So yeah, with today's tools, you can go in and figure out where the genetic changes are. And we and others do that all the time.
So that's genetics too, even
That's
though you
genetics too. But keep in mind, spontaneous immortalized cells have been used forever in pharmaceutical production,
Sure.
in other kinds of foods, et cetera. It's a pretty standard, right? And regulatory wise, like I said, there's little oversight.
But is every time a cell immortalizes spontaneously, is it the same thing?
Nope.
No.
No.
So that seems very bizarre from a regulatory standpoint that some kind of a mutation that makes them immortal is more acceptable than something that you precisely do with a CRISPR.
I agree, and if I could jump in.
Please.
Since I worked on this sort of that exact question when I was at FDA. I agree with you.
Yeah,
I would want precision over, but FDA operates on predictability and familiarity. So the phenotype of this spontaneous immortalization is very predictable, and it is predominantly very also consistent. And because consistency and predictability equal generally a safe production process, I'm very familiar with it.
okay.
But I agree, the modification is actually much more preferred.
So we're very good points. I agree. Yeah.
Related question, what causes the spontaneous immortalization? Why?
Sometimes we just stress the cells, right?
Yeah,
We take them off their media and they adjust. You're just like your own cells can spontaneously mutate, right? You have control systems in your cells that fix things, but they don't always fix everything. Sometimes UV light causes some chain scission in your DNA and that causes some rearrangement. So it can be any stress to the cells that's not your steady state conditions. Even under steady state, you go through mutations. This is Darwinian evolution at its best, right? You're going to have mutations, selection is going to happen, and you go forward. So there are now companies, again, not to go off on too many tangents because I'll never get through my talk, but there are companies now dedicated to just cell line development based on these techniques, right? That's all they do. Company gives them a cell that says, we want our cell to grow and double twice as fast as it does, right? Because quicker doubling means shorter times in culture means less cost, right? And you get more cells per unit volume quicker. So their company's just dedicated to this. But I agree with Eric. We have to make sure everything's safe, and that's part of the whole.
I'll say the cheaper option is to spontaneously immortalize,
Really?
but and more generally, but the upside product is intentionally genetically engineered, precisely trying to make that point a little bit more common.
And we really like CRISPR because you can really go in and know exactly where and what's changed. So I think that's going to be the rule going forward, pretty much. Yes, please.
Any evidence to suggest that this will make a contribution to longevity, to make sure that everybody lives in a blue zone, so that we can assure that we'll have 12 billion people on the planet instead of just 10.
I'm not sure we want 12 billion. That's another discussion. The answer is maybe, and I'll talk to that in a little bit if I don't run out of time, because I think there are some opportunities. I'll just call it healthier eating, or I call it personalized foods, therapeutic foods, where yes, you could probably improve longevity, whatever that means, but I don't want to overpromise there.
Thank you.
Yes, please.
It's a quick question from a bioprocessing, biomanufacturing standpoint. So as you mentioned, right, a lot has been developed in the context of immortalizing cells, but mostly the product has been therapeutic proteins, let's say. But now the product are cells. I'm just curious as to what has to change from a bioprocessing, biomanufacturing standpoint to ensure the genetic integrity of these cells.
Cells are safe, yeah. Most of our lead comes from actually stem cell biology, right? People are using cells therapeutically, right? Injecting everything in the brain to the body, et cetera. So there's a lot of precedence here, right? Because there's a lot of genetically edited cells being used that way. And there's no evidence of any issue there. It doesn't matter. We have to be vigilant. We have to run all the proper food safety tests with these products, and they have to be vetted over time. We have a current project ongoing, just looking at these cells in storage, right? Do they change it over time? Just so we have the data, because the data doesn't exist. So a lot of work to do.
I can quickly answer. One, we take pharma, we do master cell banking specifications. One, so we borrow straight from pharma because it's the best system. And two, the safety assessment requires specifications be set for phenotype. So you would reject lots if it doesn't fit a certain phenotype.
So it's not the wild, wild west, but the regulations have to follow, yes.
This is a real quick question. Are CRISPR-Indels still regulated or not regulated? Or I know obviously you insert a whole gene with CRISPR, it's regulated, but my understanding was CRISPR-Indels were not currently considered GMOs.
As far as I know, that's correct. You would know better than me. I don't think it's changed.
If you intentionally alter the genome of an animal or animal cell, it is considered genetically engineered currently. That's the big debate, is the intention. So yeah, that it's not about the actual science. That's where we're going to talk about this later.
All right, let's get back on. All right, back on track. It's all right on the clock. All right.
It's a very good question.
Okay, all great questions. Keep those in mind for our further discussions. All right, so very quickly, I started a little bit on the left about cells, so you had a sense. Middle now, I just want to talk about media, right? We got to grow the cells. Every techno-economic analysis, every life cycle analysis that you read anywhere says media costs are the biggest challenge for the field. That's where the major costs are. And so the biggest things you want to think about in all the media are, one is the serum, to try and rid the media of serum because it's animal derived and you're trying to grow animal-free products. And the major one are growth factors. These are proteins, sustainable proteins, recombinant proteins, and they're very expensive, right? So how do we get there? So just a couple of slides from what we do. Other labs are doing their thing, right? I can tell you in the course of 3 years, we went from almost $200 a liter in media costs to grow our cells to now, I have another slide after this, down to $15, $17 a liter. That's just one lab, right? Because now we're focused on this. And how do we do it? One is we got rid of the serum. We used what we call a recombinant albumin. And albumin is a protein, sustainable protein you can produce recombinantly. And it basically has the same function as bovine serum albumin. So that brought us way down. And then we found we could get rid of that recombinant albumin, which is still very expensive, by finding a very inexpensive agriculture plant product waste. This is one of the oilseed crops. We had a very crude fraction, and it replaces the use of albumin. And that's where we went down again over there. So when people ask me, I don't think costs are going to be the driver in the long run. In the next 3 to 5 years, you'll see costs really, really down, way down, right? We still need supply chains and everything to feed the process. But this isn't the issue. And the paper we just published literally last week was to even get rid of the growth factors, right? So now we're plummeting below $20. We're down to, I think this is down to like less than $18 a liter to grow them. I think this will come down another order of magnitude over the next 3 years. And we did this by, we call it self-priming cells. So we took our bovine muscle cells, and now we have those cells make their own growth factors. So you don't need to add them. They're just self-priming at that point. and they work, right? There's still work to do, but the point is, these are all techniques coming along. All right, and then we're using a lot of AI and other methods to develop further media optimization, which is going to further bring down the cost, just to overstep that. So again, costs are still an issue, but I think they're gradually dissipating. I think the biggest issue is scale, and for time, I don't want to spend more.
Don't worry about time.
Really?
I can buffer later. It's okay.
Be careful what you. I appreciate that. I'll buy you a beer tonight. So I think everyone knows, the amount of meat produced in the US and around the world is just unfathomous, so high, right? 500 million tons, et cetera, right? So if people have done calculations, right, if you look at the entire bioreactor volume available on the planet to host cells to grow meat of the future the way we're talking about, we're orders of magnitude. That scale up capability does not exist on the planet. And so the question is how to fill that void. And so we need lots of efforts to look at scaling. Number one, what are the next levels to go to? And we need lots of public support, government funding to build the infrastructure to do this. It does not exist today. So to me, that's the holy grail we need to get to in terms of bringing this field forward. For now, we can do a little bit. We can get enough to put in a restaurant, great, but it's not going to be on supermarkets till you can start to address this problem. Now, the other thing I will just take issue with, and I don't think Eric necessarily agrees with me on this one, is most of the startup companies in the space learn from pharma, right? And so you see what pharma does on the right, big steel tank, and really the tank is the small part. It's all the plumbing, process control, all the things you need to do to get your cells to behave, right? Plumbing, process control means cost, right? I just said cost is the driver here. So the more you're going to go to large steel tanks, the more you're going to have to fight the costs going forward. And I think that's going to be a problem. So I think it'll solve certain problems, but we need innovation, is my point. You need to really think outside the box of how to grow cells at volume that you can't do today. So that's where we come in with our USDA program. And I'm not saying we're right, I'm just telling you what we're doing, and you can laugh at this or not, right? So this is what people do today. We argue that we need to look at systems that are not constrained by traditional mass transfer, things you know all about. We need less process control, and we need both continuous and modular design. So you can make this in a house, to a small community, to a city, doesn't matter, right? It's kind of a universal concept. So we are working in all these areas. I'm not going to go through them all today. I just picked one to show you the craziness of our ideas. So this is what we're trying to do. We're trying to literally knit meat, okay? And it may sound a little silly, but the idea is to create texture and dense meat, one of the goals here, in a different way. And so this is what we're doing. And I will tell you, we've already gone through this whole process, and I will tell you it works. It's not scale, I'm just saying it's research, right? But we simply feed our cells, I've talked about the cells, onto an edible fiber. So we call this 1D food production. And that's the only place you need to worry about growing the cells out till they make a muscle or fat. The rest is all just traditional textile engineering, right? Weaving, yarning, whatever you want to do, knitting, et cetera. And so we borrow from established infrastructure, established devices, and scale up. We don't have to reinvent all that. You just have to do it in a food context as opposed to a traditional textile process. So this is what we're working on. Thanks to Merck, they funded a 3-year program with us to get this to the scale we have. And now we're busy trying to sort of spin that out of the left part of the pot, spin it out, into a real viable startup. So we'll see how that goes. But we're doing the same thing on our 2D, think of roll-to-roll manufacturing as opposed to, large-scale bioreactors and so on. You still have to make the cells and all that product, but the rest gives you dense products when you're done. All right, last bit on the right, where do we get to the end? So just materials, we need materials for every part of the process. The feed, the media, the scaffolding, carriers for cells, all this rides the process. In the lab, we do traditional cell culture, which many of you in the room know about, which is take our favorite sustainable protein. We mostly use things like soy protein, zein, glutenin, pea proteins, and then we use some polysaccharides as well, like chitosan and so on. We grow the cells under different conditions in terms of muscle and fat and see how they do to optimize the interface to get things moving forward. One of the things we just published is to try and get people out of that mindset and try and develop new methods for people to follow for the field. This is one I'm pretty passionate about, is to say it's not a time-stamped process, right? Because if you look at time on the x-axis and mass, biomass, cell mass on the y, that's the nutrition, the taste, the flavor, right? They change over time as you grow those cells out on your edible scaffolds. So you have to decide what you're trying to produce, right? What mechanics you want, how much cell, what nutrition, et cetera. And then you go in with these models and you say, okay, I think I have to grow it for 7 days, then I harvest, and then I have what I need. So the idea is to build this into a paradigm you can start to use to predict early on. So I call it a sort of the next generation mass balance approach to really understand the dynamics of the scaffold, the cells, and the new matrix the cells produce to make all that work together. So that's ongoing work. And then we do nutritional quality, safety, et cetera. So this is my attempt at flavor and taste, right? So the big question we had in the group after 2 years of doing this was, if we grow pig fat from pig cells separated from the animal, will they have anything the way you would think of pig fat when it's cooked that you're used to, right? Would it have the flavor? Would it have the taste? We had no idea. And it's hard to find any literature that covers this. So the question is, right, the cow we're used to, we know what to expect, and consumers will want something they know. But if we isolated the cells we talked about and do that, what happens? So this is what we do. Right here are the cells growing. These are the fat cells from the pig, just so you see what it looks like. We scrape them up. You can see little vials of fat cells. And then we have a little cooker thing in the lab, and we had to get IRB approval to do all this with the students, right? And then we have, first we do all our traditional detailed analytics of the fats. So here's the regular in vivo fat from the pig that we got in the butcher shop. And on the right-hand side are the volatile compounds produced from our in vitro lab grown fat, right? And again, I don't have time, it's all published and another one's coming out. We track all this and they're not that different. There are differences, but they're not that different. And ultimately, those are our students doing the tasting or the smelling. And the answer, and all this is getting published as well, is that there's not a lot of difference. Thank goodness, right? It's like, there are differences, don't get me wrong, but they're not significant differences. And the beautiful thing here is if you follow what I said, we can start to fill in if there's something missing, we know what to do, right? We know what to feed those cells to sort of bulk up that particular flavor or taste. So this is ongoing. That's as far as we've gone so far. The goal is next to do taste testing. We're just getting to that approval as well, but that's kind of next on the docket. All right, let me just close. Man, I apologize, I'm still.
You're good.
Okay, so I want you to think now much more broadly. You know, you're used to your foods for 10, 20, 30, 40, 50, maybe 60 years, right? But think of the future. The future is now completely divorced from the way we're traditionally making food. If you think about opportunities from what I talked about. One is, you're not confined to the 12 or so species you're used to eating, you know, like cow, pig, chicken, duck, certain fishes, right? Think about expanding your palate to literally any animal on the planet where you could harvest cells and carry out the same process I talked about. There's nothing preventing that. And so you could start to expand palates that way. Number two is therapeutic foods. I'll show you one quick example of what we're doing there, how to take these foods to the next level based on the question earlier. And that gets to personalized foods and then making food fun again, right? Not just family gathering to cook and eat, but family gathering to make the food and then cook. Right? I think lots of things to think about. Here's one example of what we've done. This is published, right? And it really works well. We took our cow cells and we fused them genetically with carrot cells, but really the pathway, synthetic biology, was to make vitamin A. And so this is the details, it's all, like I said, published, but we could actually make our cells, our cow muscle cells full of vitamin A. And so you say, Dave, that's crazy. Why do you want to do it? And you want to do it for two reasons. One, the lipid oxidation becomes reduced, even just during storage of the raw cells. So that means the food is healthier, longer in a refrigerator or on the shelf, number one, so better for you. And number two, when you consume it and cook it, it's also more enriched in vitamin A. If you just added vitamin A, it's not as stable when you just add it. And so that means if you're somebody who suffers from gut health issues like IBD, irritable bowel disease, our goal was, would this help the gut system at all by having a food that's enhanced in these features. And those are ongoing studies to determine the effect on gut health. But this is just one of, we probably have a dozen projects going where we're engineering the cells to accomplish specific therapeutic goals at the same time. And then I mentioned you could start to really go crazy, right? Any species, you're not going to hurt the species, right? So it's one-time biopsy, and then the species goes free, and you can grow. So, for example, we work on insect cells in our lab because we see their cost of production is so much lower than these animal cells we're talking about, and yet the flavor, taste, nutrition is just as good, if not better, than traditional cells. So lots of ideas here. Happy to chat about these. So let me close really quickly so I don't take too much extra time. What we're doing sort of logistically in the program, so you're aware of it, and we're looking for, obviously always looking for ways to collaborate with others like the group here. One is we have very active education programs. We now have graduate education, what do you call it? Certificate programs. So we have a cluster of classes on cell ag. We have undergraduate minor degrees in cellular agriculture. And just so you understand this, we cannot keep up. Right, we have more student demand if all these courses than we can keep up. There's so much interest in these courses. So I hope that will evolve here as well. We have lots of collaborations with industry, other academic labs. You can see our 8 academic partners on our large USDA National Institute for Cellular Agriculture. Hopefully we'll be adding, Arkansas here as well. If that works out, that'd be great. And then we keep innovating. We file patents, publish. We do everything to be transparent. So everything gets published, peer review, so it's available to everybody. We host, so anybody here, we host students and faculty all the time from all around the world in our labs to learn whatever we're doing. So please, if there's opportunities and there's a need, we'd welcome folks to come to Boston. Just visually, so here's a map of the academic collaborator network through our USDA program in the US. There's a gap in Arkansas, but hopefully not for long. But we're countrywide, and this is around the world. We collaborate with almost all the major activities in this space around the world. Also industry partners all around the world, very active with those folks as well. If you care, we cover, and I've covered this already today, from education, media. I didn't talk about TEA, environmental sustainability issues. We have a large effort on looking at the data we generate and how that fits into sustainability and many other factors here. We work directly with industry in a couple of different ways. I'll just highlight this way, which we talked about at dinner, which is, or yesterday, is we have a series of partners that pay an annual fee, and industry tells us what they need. So it's very different than our traditional grants, right? Where we tell the world what we're going to do. Here, industry gets together as a team. They give us a list of, okay, Dave, we need you guys to solve these problems for us. And these are kind of pre-competitive problems is the idea, right? So it's not owned by one company. And then we assign faculty that want to work on these to the different projects. They get the funding that industry pays, and we go off and do it. And that's the way it's worked so far. So happy to talk about those models if helpful. So this is our overall program. We have our academic program, education, et cetera. We have our institute from the Department of Agriculture, our industry consortium, and then we just spun out a commercialization lab to help move things out. And let me just close by saying, these are my amazing cell ag students. You can actually see my daughter hidden in the back there at one of our early lunches. And USDA and our nonprofits are the main supporters, some DOD, ARPA funding, some NSF funding, so thank you to them. And then this is my larger group that works on the biomaterials, sustainable proteins, tissue engineering, with lots of collaborations all over the world. That's it, thank you. Sorry to go a little long.
That was a fantastic primer and really whet our appetites. You have heard mention of Eric several times in between. He's going to come to with the Q&A panel later, and I think he had to take some calls, so he has to step out. But let me just give you a little bit of background on who we have brought in also, and he'll be doing more for the panel part in the afternoon. But he's a professional molecular biologist, genetic engineer, startup executive, and former federal biotech regulator. So he was he's currently CEO of a company that he has called Good Human Strategy and Design Agency. But before that, he was the vice president for Upside Foods. So, he took that whole thing through the whole process. So, what the regulatory policy should be, linking up with government affairs. So he comes with a lot of experience in that space. And even before that, he was linked in before the private sector with the US FDA and looking at this portfolio of, the food in the FDA part. So that food part, and has also been, this entire background of government, regulatory and industry background, and actually getting this all protein really commercially, now approved in the process, getting that whole process put in place. He comes with that huge experience. So he's going to help with asking more questions and then leading the panel in the afternoon and going through this. Now let me introduce Dr. Sandberg.
Oh boy, good morning, everyone. Difficult, interesting task to follow the talk of Dr. Kaplan. But the reason that I gave him some of my time was like strategic, because some of this, there is an overlap, and my goal is actually parallel path and see possible collaborations between ag, cellular agriculture, and microprotein, which we'll be talking a little bit today. So yeah, so we'll talk a little bit about what the context, how we fit in this world. Talk a little bit about microprotein and how we can coordinate and find synergies with other alternatives for a sustainable future. So as Dr. Kaplan presented today, there is an increase on demand of animal protein. And when we put in dollars, which one is there? When we put, so we have also not only increase on demand on consumption, but also the value added to it, right? So the market is increasing rapidly. But at the same time, to meet that demand can be challenging sometimes from different points. So for example, when we think about a 1 ounce steak, the carbon emission equivalent associated for the production of that steak means 5 trees will have mature trees will be working during 1 year to neutralize that for 1 steak. Okay. So when we think about, okay, we have an increase on demand, but at the same time, we're going to a trend on decreasing forestations, natural water resources, what are important also for our, to decrease the carbon emitted, we can find ourselves in a conundrum or like that. So, but I'm assuming almost every one of us already kind of agree on that and are on the same page on that. So can we meet the protein demand in an economically, socially, and environmentally sustainable manner? And that's, I think, is a really important discussion that we have to put on the table. We have to make sure we are tackling the environmental point on protecting our environment and thinking about nature, humankind, but also we have to understand that we live in a world with an economic system that must be sustainable as well. And added to that, the equity, the social component to that all should be closing the equation to make sure we are not leaving anyone behind. So just strategically, as was mentioned, I'll just go past this. I won't take so much time. The plant proteins did a lot of heavy lifting on entering the sustainable protein world. And that's personally, I'm really grateful and I'm respectful of that, because it was a tip of a spear on entering the market, right? Like the proteins, the high moisture, the expanded proteins, the beverages that we have today, as was mentioned a few years ago, were not even spoken of. And they opened the concept, they planted the seed on the consumer market, on consumers, about an alternative way to get protein. And this is really important. We have to be really conscious of that. But then when we look into our future, we need alternatives to still fill that gap, right? That's pretty much where I'm coming from. And all of us are working on this consortium. So we talked about the cell agriculture, the cultivated meat. Dr. Kaplan talked about the precision fermentation, right? There are other players in the field with air protein, for example, where they capture air, use air to feed reactors to produce protein. So there are a lot of alternatives there. But also for me, what's very important, we can't forget the farming and agricultural practice. We have also, going back to your question on that, I don't think that's my point of view. They are not mutually exclusive initiatives. We have to still work on sustainable farming and agricultural practice to make sure we are going in a parallel path. And it is possible. It is possible. And that having everyone together in the same room talking about it is one of the first steps to it. And it's happening. And I hope we can foster that conversation. What I'll bring here to us today, on a kind of a high level to discuss and open to a discussion, is the concept of mycoprotein. When we think about mycoprotein, we think about fungus, like a protein coming from fungus. And I bring kind of future, present, and past because it's been around since the 70s. It's not necessarily a new trend, but it is gaining traction also with the movement that we're going through towards alternative sustainable proteins. So when we think about mycoprotein, we're talking about some specific strains of fungus that will be grown, can be grown in a tank, but also it's pretty similar to what we see, for example, when we see a mushroom growing in a lawn, for example, when we're going for a hike. The same concept. What we do, we harvest that fungus growing, take out of the water, and then we can either use as a flour, as an ingredient, or we can actually texturize as an alternative. That's the concept. So when we think about how this looks like, I won't go too much into detail on this, but we are talking about a product that has about 40 to 50% protein. And what kind of protein are we talking about? It is a good quality of protein? Yes, we're talking about protein that is comparable to some extent to casein, which is one of the gold standards when we talk about protein digestibility. So it is a good opportunity there. How it is made, how we produce that, and that's where we're going to be using this as a connection with the agricultural cellular agriculture. Sorry. Pretty much you have your raw materials, your services, your utilities. You use a fermenter. When you reach steady state, you start to produce your microproteins, and you harvest in a continuous state. So you have a fermentation cycle, let's say between 4 to 6 weeks, and then you're feeding, harvesting, feeding, harvesting, feeding, harvest. Once you have your protein, your biomass, you separate it from the water, and then you can either process to texturize as a meat, or you can use as an ingredient for other applications. So corn, one of the players I'll mention here, that started this movement in the 70s in the UK, they say that they produce the weight of a cow on their bioreactors every hour, considering protein production. So that's kind of the scale that commercially available. So we have corn, one of the starting figure player on the field, started in the 70s, really established in Europe, in UK. Here in Colorado, we have MITI, which also is doing a lot of work on the mycoproteins. Cargill is working with enough food. This is kind of what I got last week when I was doing some, they had a lot of announcements on this, on the marketing commercial level with their Abunda ingredient. And there are other players. So this is our focus on submerged fermentation, but we have also some players on solid state fermentation to produce microproteins for human consumption. And then when we think about the synergies, the conversations that we have regarding cellular agriculture and microprotein, I remember attending some webinars and like when planning and thinking about it. And one of the common trends from the engineering point of view is that scalability of production. How we can ensure that is the pharmaceutical technology can be brought to a large scale food production technology. We need to have the knowledge, the know-how, the infrastructure, companies that are able in tackling that field, workforce training. So there's a lot of components there. There's like this white space that must be filled. And the way that I personally see as the microprotein, how it integrates with that is helping to develop the bioreactor design, operation, training in several segments to support a future larger scale of cellular agriculture. So there's a lot of synergies, potential synergies there that they will extrapolate to reaching that goal. When we think about also agricultural farming, so we have to feed those fungus for them to grow. So can we work towards cycles where I'm utilizing agricultural byproducts, waste, industrial waste, treated properly to make sure we are meeting all the regulatory aspects, which is a different conversation because we have similar technology. For example, when we talk about biofuels, right? We use a lot of industrial agricultural byproducts for biofuels, but how can we, what is the regulatory aspect to make sure that waste being processed by the fungus producing a product meet human standards for consumption? So there's a lot of conversation that we have to make, but there are a lot of possibilities there that we can discuss. And when we think about the microprotein application, this is just a few examples that we have. We have several works done with rice straws, byproducts from production, corn, byproducts from beer, from wine production, sea lettuce, that was really interesting. So there are different applications looking for the region of Arkansas, other than rice and corn, sorghum. So can we integrate that, having this processing facilities integrated with agricultural system? Because personally, the way that I see those systems should be integrated, should be alternatives, parallel paths going together. And there are a lot of opportunities there when we look into it. So just like as a closing, can we, going back to the question, can we meet the demand of the protein demand economically, socially, environmentally, in an environmental matter? So I don't think there is a silver bullet. And if we look just for one alternative, we may be missing a lot of different alternatives. So can we work together to several fronts just to meet several consumer needs, demands, to make sure we are working together and not leaving anyone behind? I think that's what we should be looking for, right? A synergistic alternatives, but always following a science-based approach. We have to make sure we have like a proper and solid science-based solutions to complicated problems. And I believe with that, we can achieve, an economical, social, and environmental solutions for this complicated problem that we have. So this is kind of the way that I took this presentation, was more a little bit on a higher level. I don't know, I'm not sure how much of us is familiar with the microprotein and how it's entering the market in US. And as we go throughout the day, I'm open to discuss and have more conversations on it. So I think that was it. Thank you very much.